Assalamu alaikum. I'm Dr. Sabira Dkhar, demonstrator in the Department of Community Medicine, GMC, Srinagar. Today's topic is genetics, part 1, and before we go into the presentation, we'll have a look into the video. Importance of genetics. Genetics deals with molecular structure and functions of gene. It talks about gene behavior in an organism, the pattern of inheritance, and gene distribution. The variation and change in population due to genetics. The understanding of the cause of diseases and how individual responds to therapies. Genetic disorders can be grouped into chromosomal, unifactorial, or multifactorial. Sex disorders, like the Klinefelter syndromes in males, where 2 or more X chromosomes are seen, example XXY or XXXY. XYY syndrome, also seen in males where you have an extra Y. Turner syndrome, which is seen in females, the XO composition where there is a missing chromosome. Superfemales are where you have 3 or 5 X chromosomes. Unifactorial or the single gene or also known as mendelian disorders. They are single gene disease which occurs due to a change in the gene that leads to a loss or disturbed function of the gene. It can be autosomal dominant, like the example of retinoblastoma ABO blood group, autosomal recessive, like albinism or phenylketonuria, sex-linked recessive, like hemophilia and G6PD deficiencies, sex-linked dominant, like vitamin D rickets. Multifactorial. These are known because of multiple reasons for a disease. Multifactorial disease. Other diseases like erythroblastosis fetalis, blood diseases, sickle cell anemia, thalassemia, and cystic fibrosis. Advances in molecular genetics are DNA technology, gene therapy, Human Genome Project, Human Genome Diversity Project. DNA technology. It involves the synthesis of DNA probes, DNA sequencing, polymerase chain reaction, and restriction enzyme. DNA technology examples are seen in insulin, erythropoietin. There are the cytogenetic techniques involved where insertion of a missing DNA sequence can be done or insertion of a coding DNA sequence can be done. Gene therapy. Gene therapy is when DNA is introduced into a patient to treat a genetic disease. The new DNA usually contains a functioning gene to correct the effect of a disease or causing mutation gene. Gene therapy uses a section of DNA to treat or prevent a disease. The introduction of gene sequence into the cell is done using a lipid or a virus or a receptor. The application is seen to correct a gene mutation, like in case of cystic fibrosis, or kill a cell, as in case of a cancer. Gene therapy, also called human gene transfer, is a medical field which focuses on the utilization of a therapeutic delivery of nucleic acid into a patient's cell as a drug to treat the disease. The potential medical uses are seen in gene doping, human genetic engineering, or treatment of a genetic disease. Gene doping is usually seen with athletes who may adopt gene therapy technologies to improve their performance. Human genetic engineering could be used to cure diseases, but also to change physical appearance, metabolism, and even improve physical capabilities and mental faculties, such as memory and intelligence. Treatment of genetic disease. Genetic therapy or gene therapy approaches to replace a faulty gene with a healthy gene and are being studies for treating some genetic diseases. Diseases such as sickle cell disease that are caused by autosomal recessive disorders for which a person's normal phenotype or cell function may be restored in cells that have the disease by a normal copy of that gene that is mutated. It may be a good candidate for gene therapy treatment. Human Genome Project was an international scientific research project with the goal of determining the base pairs that make up the human DNA and of identifying and mapping all of the genes of the human genome, both from a physical and functional standpoint. It remains the world's largest collaborative biological project. The planning started after the idea was picked up in 1984 by the US government, and the project was formally launched in 1990. It was a 13-year-long publicly funded project and was declared complete on April 14, 2003. The aim was at identifying and sequencing all the genes in the human genome. The attempt to systematize the research on mapping and isolating human genes, with each coding gene defined and sequenced. The main agencies involved was UNESCO, France, USA, UK, and the European Union. Applications and proposed benefits. Benefits for many fields from molecular medicines to human evolution. It helps to understand diseases in genotyping of specific viruses to direct appropriate treatment. Identification of mutations which will link to different forms of cancer. The design of medications and more accurate predictions of their effects. Advancements in forensic applied sciences, in biofuels and other energy applications, in agriculture, animal husbandry, bioprocessing, risk assessment, bioarchaeology, anthropology, and evolution. Another proposed benefit is the commercial development of genomic research related to DNA-based products, a multi-billion-dollar industry. Human Genome Diversity Project, started in 1990s by the Stanford University's Morrison Institute, with a collaboration of scientists around the world. It helped in increasing the understanding of human evolution, to understand the genetic relationship between human population, and interpret them in terms of genetic drift, natural selection, or migration. Example of this was the frequency and distribution of a rare single gene disorder which was related to history of human migration. The potential benefits of this. The HGDP data has been used in studies of evolution and expansion of modern human population. Diversity research is relevant in various fields of study ranging from disease surveillance to anthropology. The genome-wide association studies try to associate a genetic mutation with a disease. The diversity research could help explain why certain ethnic populations are vulnerable to or resistant to certain diseases and how populations have adapted to vulnerabilities. I hope you have understood this part of genetics. Thank you.